All right, well, I also have a fish story to tell, fish and foam. Today I'm going to talk about a study that Woodard and Karn conducted that looked at impacts to a river in Connecticut related to a release of aqueous film-forming foam, or AFFF. And we looked at how PFAS from that release moved in that river over time, and as well as in different media. So in June 2019, there was a malfunction of a fire suppression system at a hangar at Bradley International Airport. And an unknown volume of an AFFF and water mixture was released, approximately 40,000 gallons, we believe, and we think of that approximately 1,500 gallons of concentrated AFFF. We know for a fact exactly what AFFF the formula was. It was, in this picture here, the 3M, 3% concentrate. And this release moved into a storm drain and ultimately went through the storm drain to a wastewater treatment plant, which discharged to the Farmington River, the lower Farmington River. And so we conducted a number of emergency response actions and started our investigation trying to figure out what was going on in the river. And then unfortunately in October of that year, while we were undertaking our assessment, there was a second release of AFFF. And this was related to an airplane crash at the airport, sort of a tragic crash of an antique B-17 bomber. And that resulted in a release of about 25,000 gallons of the AFFF water mixture from an unidentified AFFF source. And that entered Rainbow Brook, which is a tributary of the Farmington River. So we had these two distinct releases. So today I'm going to talk about our overall assessment. We conducted an assessment over approximately 18 months. Our purposes were to really look at how PFAS moved in the river, what media PFAS compartmentalized in, how far did those impacts extend, and how did those impacts change over time? And ultimately, our data was used by the Connecticut Department of Environmental Protection to inform their recreational advisories for the river. The river is a Class B waterway, so it's used for fishing and swimming and boating, so not as a drinking water source. So here's an overview of the investigation area. Up in the top, we have the airport. You can see there, the green line represents the release from the hangar, the initial June event. And that green line represents the storm drain that basically discharges out at a wastewater treatment plant into the river. And then the blue line up near the top represents the path that the B-17 crash site release entered through Rainbow Brook into the Farmington River. So you can see a few other features here. Upstream of this, we have Rainbow Reservoir, and that's used as our control or our reference area. So this is upstream of that, either the Rainbow Brook or the outfall site. And then we have what we refer to as a contamination zone, which is the area within the immediate vicinity of the outfall. And then a downstream segment, which is approximately 3 miles downstream, closer to the confluence with the Connecticut River that you can see here on the right. So ultimately, we collected over 90 samples from 6 different media, including AFFF product, various components of the wastewater treatment plant, biosolids and wastewater, surface water, sediment, and fish fillet. I'm just going to focus on surface water and fish fillet. Those are the 2 media for which we have multiple rounds of data. So that will be the main focus of my talk. And for surface water, we had grab samples over 6 different rounds between 2019 and 2020. For fish, we evaluated 2 different species, yellow perch and white sucker. And we looked at fish fillets that were descaled, but the skin was left on, and we took composites. So what did we find? For surface water, PFOS was detected at the highest concentrations overall, and I guess that's not really a surprise, given that it was mainly a PFOS release. So the chart shows the sum of the concentrations of five PFAS compounds that are currently regulated in Connecticut, and I refer to them as the PFAS 5, and that's PFOA, PFOS, PFNA, PFHPA, and PFHXS. You can see the lines here look at both time and distance. So the red line is the concentration of the PFAS 5 following the initial release. Orange is the round subsequent to that initial release. The yellow line is the represents the samples that were collected after the B-17 crash. And the blue is our November round, collected only at a subset of our sampling locations. So this light green bar you see at the bottom represents our background conditions, so those concentrations that we got from Rainbow Reservoir upstream of the site. And so you can see there are changes over time as well as distance. After the first release, we see an approximately 100-fold decrease about half-mile downstream of the outfall. And we also see a substantial decrease in concentrations just a day after the B-17 crash. So this makes sense. It's a river and the water moves. And so by November, the concentrations that we were seeing in the river were fairly consistent with those that we were seeing in reference areas. So another way we can look at the data, apart from just concentration, is by looking at the composition of the other PFAS that are present in a sample. And so, we know AFFF and our background surface water concentrations had very different composition. Here I show a radar plot. So this is a kind of a nice way of looking at the data. This shows the 7 most common PFAS compounds that were detected in our AFFF product in our background samples. And so what you're seeing here is basically a percentage of the total of the 7 PFAS concentrations plotted on a log scale. And the red line here is the AFFF product signature, and the purple line shows our background signature for our background samples. So you see they're quite different. When you look at radar plots for all the different rounds of samples, you can really see that there is a change over time. So prior to the B-17 crash release, I'm sorry, these are all prior to the B-17 crash release. So you can see that, collecting samples just a day after the release or a couple of days after the release, this bright red line really mirrors what we saw for the AFFF product. And over time, several months later, by September, you're seeing a pattern that's very similar to what we see for background. So remember, background's that purple line. So we can really see that that does change over time. For fish sampling, like I said, we looked at 2 different types of species. And one of the reasons we did that is we wanted to look at a bottom dweller, so a fish that tends to spend a lot of its time near the bottom of the water column, and then a yellow perch, which is a small predatory fish. And I think has been discussed in some of these other talks today that, PFOS is known to biomagnify. So we do tend to see higher concentrations in different organisms that are higher up in the food chain. And so what did we find? So for our chart here shows basically PFOS concentrations over time in both perch and sucker from both the upstream and the contamination zones. The green lines represent our reference samples from Rainbow Reservoir, and the blue lines represent the contamination zone samples collected at and downstream of the outfall. A couple other lines here. We have an orange dashed line, and that represents a baseline concentration that we obtained from an EPA 2009 National Rivers and Stream Assessment Study. And that we found data for perch fillet samples from the Farmington River. So based on that, we had an average concentration of about 26 nanograms per gram. So that provides us good historic baseline, as well as the reference data that we collected from upstream. So what you can see here is that, oh, I'm sorry, there's two other lines I meant to point out here. We have the Connecticut fish advisory levels. The red line is a do not eat level of 159 nanograms per gram, and then the 20 nanograms per gram is an unlimited. So basically there's, below that number, there's no limits recommended for fish consumption. These are important because what we saw in our initial July 2019 round was that the perch concentrations were above this do not eat level. And so because of this, Connecticut issued a do not eat advisory for fish from the lower Farmington River. And as you can see, perch had the highest level of PFOS detected in fillet samples. So as discussed, the perch is a predatory species, so we would expect that to be higher than what we saw in sucker. So the data do kind of jibe with that. Slides keep changing themselves. And then we do see concentrations decrease fairly rapidly. So, overall, we saw PFAS concentrations decrease to reference level concentrations within a year after the release. So that was pretty significant. And so this reduction, I think, is consistent with other studies that show half-life of PFAS in fish being in the order of several weeks to several years. Now, remember, we did have a secondary release, and there are always upstream sources of PFAS that are coming downstream. So it's a little, we can't really estimate a half-life based on this data set, but it is kind of interesting to note some parallels there. It is also interesting to note that sucker and perch concentrations upstream remain pretty consistent over time, where we saw a steady decrease in the contamination zone. And so PFOS concentrations really swamped the data set. So those were definitely detected, that was definitely detected the highest concentration in both species, but we did have other contaminants, other PFAS contaminants as well in our samples. So over in this inset box over here, you can see there's a whole host of other contaminants detected. And generally, we see this enrichment of carbon 10 to carbon 14 perfluoroalkyl carboxylates in fish tissue. And you can see some patterns in this as well. So generally, we saw similar patterns for sucker as we did for perch. So I guess not that surprising. There were some slightly different some patterns there, but overwhelmingly it's pretty much the same type of thing. And so here again, we're using radar plots to visualize how PFAS signature changed over time in fish tissue. The red line shows the contamination zone samples in July 2019, just after the initial release, and the green line shows the results for reference samples. The yellow and blue lines show fish results 3 months and 1 year post-release, respectively. So you can see the signature of the 2019 samples and the sucker are more heavily weighted toward PFOS, consistent with our AFFF release, whereas the later samples have a greater distribution of other PFAS, consistent with reference. So, so with that, so we show that, we have definite patterns of PFAS and other PFAS compound signatures in water following an AFFF release. And that rapid response was really critical in reducing those initial concentrations. But then we do see a rapid decrease in PFAS concentrations in the river in both surface water and fish tissue. And so ultimately, because PFAS concentrations were consistent with reference a year later, that fishing advisory for PFAS was lifted. And again, I think this study really highlights the importance of collecting a good background data set to really understand what your background load is in a riverway. So with that, thank you.
Okay, here's a question. Were you able to do any total fluorine analysis on the surface water? Did you sample sediment to see if it's absorbing to sediment particles, seasonal changes?
So we did do sediment sampling. I didn't present it in this study, but we had a hard time finding sediment in the river. First of all, it was very hard substrate. But what we found in the sediment, it was actually very low concentrations, which was surprising. So really on, low part per billion to maybe up to a part per million at the very highest. With regard to other analyses, we did do a top analysis. I don't have those results handy to talk about those, but we did see that there were definitely precursors in the mix, in that you would end up with slightly higher concentrations.